start us off and say thank you to everyone for joining us today for this second class in the climate change and sagebrush training series. The second class is going to be about climate change vulnerability of sagebrush-associated wildlife. And we have three great speakers today. Lindsay Thurman with the USGS, Northwest Climate Adaptation Science Center, Janet Recklow with the University of Idaho, and Cam Aldridge with USGS Fort Collins Science Center. And I will let you all know that we will have our third virtual class on April 20th. So if you can, please be sure to join that one. And there is a recording of our first virtual class. So if this is your first, I will drop a link to our website so you can register for the third and see the recording of the first one. And without further ado, I'm going to pass it over to Lindsay to share her screen and start presenting. I will be putting in some more information about the speakers into the chat so that you can learn more about them.
Just want to confirm you can see my slide and not presenter view, right? Okay, great.
It looks good.
Thank you. Yeah, thank you for having us and for giving us this opportunity to present on climate change vulnerability of sagebrush-associated wildlife. As Elise mentioned, this is a part of our virtual classroom series for the climate training. And on behalf of my co-presenters, I'd like to say welcome. And we have a quick, let's see, click on the screen here. Quick roadmap of what we're going to cover today in this webinar. We're first going to go through some examples that highlight the different ways in which climate change can impact species and result in vulnerability. We have some great examples from the sagebrush ecosystem to share with you. And then we'll transition into talking about how we measure those responses and how we use that information in climate change vulnerability assessments, which are a very useful tool for integrating vulnerability science into conservation and management. And then at the end, both Cam and Janet have some awesome case studies to share with you that sort of showcase like this whole process. And at any point, if you have any questions, please put them in the chat, if that's my understanding of our process here. And we do have a couple questions to poll you all, and I think Emma, you have control of those.
I should be able to load that for us.
Okay. Yeah, we have two questions to kind of kick off the webinar, just to get an idea of where everybody's coming from, their different perspectives and experiences.
Okay. Are you all able to see the poll?
It looks like I can see the results.
Great.
Yes, all co-presenters will see the results, and then the participants will actually see the questions.
And this information will also be helpful for us for those of you that are attending the in-person workshop.
Emma, can you just confirm that at some point we can pull this in, or are you going to share the results here in a second?
Once
I think that
everybody's done, we can share. Yeah.
All right.
Okay.
it looks like about half of the participants have responded at this point, so we'll give it just a minute or two more.
Looks like we may have reached capacity with that first question. Everybody's answered for the second question.
All right, well, I will see if I can share the results. Can you all see the results?
I can't see the short answers for the first question.
Do you see an option that says view details?
No, but just says stop sharing.
Okay, on my end, I see a view details, which will take you to a place where you can see those short answers. So I'll put that link in the chat and you can see it from there.
Okay, awesome. Thank you.
Oh, I see. Okay. All right, cool. Well, as far as the second question goes, we were interested in, for those of you that have experience working with these wildlife species, we were curious to know what kind of changes you've already observed for those species. And it looks like a lot of you have noticed a loss of habitat and increases in invasives, fuels, and fires, which we would certainly expect for this system. As well as population declines. And then some of the other options were sprinkled in there, but those were kind of the top three. And we'll be discussing all of these different factors throughout the presentation here. So we just wanted to see where everybody was at with that. So thank you. And I will continue on. So sort of a roadmap here. If you watched the first webinar, the presenters there covered sort of the physical basis for climate change. There was a great conversation about, the effects of changing hydrological cycles and temperature regimes and how that sort of cascades into, changes in the physical system, drought and fire and snowpack and moisture, et cetera. And what we're interested in here is how those physical changes are impacting wildlife species. And there's essentially two different pathways by which climate change factors can impact wildlife. And the first is sort of that direct pathway where the climate and those climate variables themselves are directly impacting organisms. So they're, causing lethal and sublethal effects on those individuals. And we'll talk about that on the next slide. The other pathway by which climate change can impact wildlife species is through these indirect effects. And that's essentially effects that are propagated through changes in the abiotic environment and the biological community itself. So these can be things like changing food web dynamics, changes in other types of species interactions, changes in the quality and availability of food and water and habitat. And so those changes that are impacted by climate factors then have indirect effects on the wildlife species themselves. And the types of responses and impacts that we're seeing are quite variable, quite diverse. They can range from changes in distribution and abundance patterns to behavior and phenology, survivorship, fecundity, fitness, immunity and health. And whether or not those changes are adaptive or maladaptive really can determine the degree of adaptive capacity for those species in the face of climate change. And we'll talk more about adaptive capacity later as well. But we just wanted to give you a frame of reference for some of the concepts we'll be presenting on for the remainder of the webinar. And I'll turn it over to Janet to share this slide.
Great, thanks so much, Elise. We wanted to just give a little bit of a overview for how it is that animals exchange heat with the environment. And this is something everybody probably remembers, but maybe hasn't thought about in quite a while. So there are four main mechanisms by which heat can be exchanged between animals and their environment. Can you guys see if I use my mouse, can you see that? No? Okay. So conduction is one that has to do with the connection, the physical connection of an animal to, thank you, to the ground. Another one, convection, has to do with the heat that's lost through a fluid. In this case, air acts as a fluid. The other of the three dry routes of heat transfer is radiant heat or thermal radiation. And we think about radiation primarily from the sun, shortwave radiation, but you see on this illustration, there's multiple other examples of radiation that's reflected back to the animal from other structures in the environment and also from the animal's body. And the last one, really important for animals as far as being able to reduce body heat, is evaporation. And that can be evaporation from the skin surface or evaporation from respiratory tissues, the nasal passages, etc. So a couple things that are really important when we think about animals in a warming climate.
It is 3:15, so we will get started on our final session.
So one is that radiant heat is really important. It can be actually the most important mechanism by which animals gain heat load in their body. The second is that really of those four mechanisms, it's really only convection that depends on air temperature. And yet when we think about climate change, we often think about an increase in ambient temperature. It's not the most important factor. The third point to remember is that evaporative cooling can be really effective and also really energy efficient as long as water is available. But for many of these dryland ecosystems, they're becoming more arid as well as becoming warmer. So that is challenging for animals. And the last point to remember is that endotherms, which we're talking about birds and mammals, we're not really going to talk about reptiles and amphibians. They relate to the environment very differently. But endotherms generate a lot of heat, and their metabolic heat that they produce themselves is actually a significant contribution to the overall heat load that gets into the equation for their heat balance in a warming environment. And so one other, sort of going back to basic principles, one other principle that affects the way that animals exchange heat with the environment is the surface to volume ratio. And you know that as animals get larger, they have a relatively smaller surface to their volume. And that's critical because animals exchange heat as a sort of as a function of their surface area, and they have the ability to generate heat sort of as a function of their volume. So small-bodied animals have a larger surface to volume ratio. It means they cool off faster in a cold climate. It also means that they can warm up faster in a warming climate. Also, small-bodied animals tend to have a relatively higher metabolic rate, so they're also generating more of that metabolic heat. In addition to size, shape also affects that surface to volume ratio. And then also, even for individual animals, their posture can affect that. This is a great example from some of Eric Beaver's work on American pikas. And you can see that that pika on the top is in a cold environment. It moves into a sphere. Actually, a sphere for any volume has the lowest surface to volume ratio. In a warming climate, animals can stretch out and expose more of their surface for heat loss. So these are just some underlying principles to think about when we talk about the way that animals exchange heat with the environment, really important in a warming environment. So Lindsay had mentioned direct effects, and we'll just really quickly go over what we mean by some of those direct effects. I think the most obvious is lethal effects, and in your poll, about 27% of you indicated that you were concerned about direct lethal effects of climate change. And there's been some really, really sad examples recently out of Australia. A few years back, in one heat wave, over 20,000 spectacled flying foxes just overheated and died. That was about a third of the population of that species. So that's a really dramatic example of a mass mortality and a lethal effect. But there are many other sublethal effects of a warming climate on animals. So, and there's just a few of them listed here. We could come up with a much longer list, but we can think about the animal's ability to regulate, to maintain homeostasis, so to regulate their body temperature and also their water balance. There are a number of different ways that an elevated body temperature, hypothermia, can influence reproduction for both males and females. There's a higher incidence of abnormal spermatogenesis when animals are warmer, including damage to the genetic material, lower fetal survival, lower lactation. There's also, for a lot of animals, as their body temperature gets higher, they reduce their forage intake, which means that they have less energy, and that's less energy to develop offspring, to grow, to deal with things like addressing challenges from parasites and pathogens. So a lot of these pathways are sort of intersecting, and very difficult, more difficult to assess, measure, quantify sublethal effects when it comes to understanding how it is that these animals are responding to climate change.
So we're going to shift now into talking a little bit about some of the indirect effects that might affect wildlife populations. I'm going to lean on an example here and throughout some of this talk on some of our work on white-tailed ptarmigan. Ptarmigan are a grouse species that inhabit high alpine systems. They're heavily affected by climate change. And much like most other grouse species like sage grouse, the young, the chicks here, rely heavily on mesic forb-rich resources that are also plentiful with insects for protein abundance, especially early in life. And from selection standpoint, we see this, that ptarmigan hens are taking their chicks into these forb-rich areas with larger insect abundances, and they're choosing to go use those. The question is, what potential consequences might climate change have indirectly affecting the habitat of a species like ptarmigan, and how they may be or may not be able to respond? And I think it was 79% of you also suggested that habitats in the poll were extremely important and concern of change due to climate change. So next one, please, Lindsay. So the next, what we've been able to look at is, with an advancing shift in climate and warming earlier in the year, we've been able to track the phenology of vegetation and insects. And this is at 3 sites across 3 different years. Sites are on the top and 3 different years down the right side. And these are individual different genuses of plants that have when their phenology curves and they peak. What we're able to do is look at these for each individual species or cumulatively, as well as with insects, and identify the peak area under the curve where the biomass is most bioavailable to a female and her chicks. And the idea is that a female should be trying to match that environment and try to have her chicks, the eggs hatch and the chicks be available to feed on those resources when they're most abundant. So the difficulty is that that is shifting over time and advancing. We'll see that in a minute. What I'm going to show you next is the consequences of the mismatch. And so what we see, daily survival is on your y-axis on the left, and we have the mismatch in days, and this could be positive or negative. So if you miss the peak by 1, 2, 3, up to 30 days, and chick age on your z-axis on the bottom right. And we see daily survival changes drastically. It falls off as that mismatch grows with a peak in phenology and availability of resources. His average is a mean of 15% lower for chick survival per day, but note that on that z-axis, it's really large drop down to as low as 15% at young chick ages. So the consequences of not having access to those food resources at a really young age basically result in brood failure and reduced survival. So this can have profound implications. So if we go look at the next slide, we're going to see that we've seen this decline over time in chicks per hen that are produced. And here's just this plotted of the mismatch in the number of days and how that chicks per hen in a population drops. And it can be halved by as long if that mismatch is 10, 20, 30 days out. So if birds miss that, we see drastic reductions in reproductive output, and we're seeing consequences of older age structure in the populations. What's really interesting is it's not quite as sad a story as we might hope, that birds are trying to match this. We've seen this advancement of median hatch day over time. You can see here plotted by about 20 days. And the start of the growing season has advanced, and birds have tried to match this. But we don't know if there's plasticity in this trait, if there's a genetic basis for it, and birds, how much birds are going to be able to adapt as climates continue to warm and the phenology of the breeding season and the plants and vegetation advances.
Okay, so we're going to do a bit of a transition here and move from talking about those direct and indirect effects to the types of responses that we're seeing across wildlife populations. So we have some more examples to show you all to do with these topics here highlighted in the orange box.
Great, thanks. So this is a figure, well, first of all, we're switching to give some examples about behavioral responses. So behavior is one of those traits that we expect animals can adjust, but we don't know the limits of their ability to adjust behavior. And this is a review article that asked the question, it was titled, "Can diurnal animals compensate for climate change by shifting to nocturnal activity?" And in this schematic here along the top, there's examples from the individual onto the community of the costs of remaining active during the day as the climate warms up. So for individuals, that's going to mean a greater evaporative water loss, onto competition for shade and water. We expect that as the climate warms and dries, there'll be less shade available, less water available. On the bottom panel, there's the cost of, for diurnal animals, of shifting their activity to be active at night. And so a number of mismatches. They may not be adapted to it. They may interact differently with other members of the same species. They may be exposed to interactions with other species, resulting in predation competition. So this is sort of a conceptual way that we think about some of the costs for either changing behavior or not changing behavior. And I'll give a little bit of an example in the next slide from some work on pygmy rabbits. So pygmy rabbits exhibit a sort of traditional mammalian pattern. The two graphs on the right, the one with the sun is their activity pattern during the daytime with this in the summer, and on the right is their activity pattern in the winter. And it's across on the x-axis across a 24-hour cycle, and it's a relative motion sensor detection. What you can see, a couple of things come out of this. One is that overall animals are more active in the summer than in the winter, but they still maintain that pattern of a period of sort of rest during the middle part of the day and peaks in activity right around the crepuscular periods. In the winter, where those red circles are at night, however, that's a period where they reduce activity at night. In the summertime, they're active throughout the night. So you can hit the slide going forward. We also modeled factors that affected activity during the night, during the summer, and that should say nighttime activity. So nighttime activity during the summer, not a surprise, is affected by the sex of the animal and also their reproductive status. But during the winter, temperature and moon phase had the largest effect on whether or not animals were active. So this is to suggest that animals have some plasticity. They exhibit plasticity between seasons, even within a season, based on temperature. So that's a little promising. But what we don't know are the consequences of shifting activity. For example, if the climate becomes much warmer, pygmy rabbits, yeah, thank you for correcting that. And pygmy rabbits need to be more active at night. We don't really know the sort of downstream consequences of that type of shift in behavior. Okay, you can switch slides. I'm going to give you another example, not from pygmy rabbits, but from another small mammal that changes behavior not by shifting when they're active, but by making use of thermal refuges. And I like this example because it is from Arctic ground squirrels. These are, high in northern Alaska on the North Slope, above the Arctic Circle. And during the summer, these animals are active, foraging on the surface. They wind up gaining a fair amount of body heat, and that's from solar radiation. There isn't any shade. They're above tree line. And once their body temperature gets up above about 39 degrees, the animals go back into burrow systems. They shed that body heat. You can advance the slides a little bit. Yep, they shed that body heat, and then they come back out and forage again on the surface. And this is a behavior that's known as shuttling thermoregulation. And so this is another way that mammals, and particularly small and medium-sized mammals or birds, have the opportunity to adjust to a warming climate. There are probably costs for this type of behavioral plasticity as well.
So we know that range shifts are likely to happen. If you're on the first webinar, you saw lots of signs talking about how sagebrush ecosystems are going to change or shift, move more northern in latitude or upslope and higher elevations because the climate envelopes are going to vary. So this is some work from my PhD student, Nick Van Lannen, where we actually modeled the abundance and distribution of various songbird species that range from the sagebrush ecosystem through the pinyon juniper sagebrush ecotone boundary up into the deeper, thicker phase 3 pinyon juniper woodlands. And you can see that the distribution of these birds does vary. And this is sort of a schematic plotting how they fall across the range of the ecotone and ecosite variations across here. As sagebrush is predicted to shift and move up those landscapes, being birds, we sort of hypothesize that they'll be able to shift or follow that transition because it's going to be fairly slow, but there's going to be consequences in that those the habitat changes. How will sagebrush get to those envelopes, and will there be transition periods where there may not be higher quality habitats as the change takes time to take place? So birds and mammals will probably shift range fairly easily, but they have to match the environment in which they've been adapted to. So there's also stresses, key responses from a physiological or a metabolic potential stresses that could happen. I'll jump back to our white-tailed ptarmigan example again, in the early 70s, Alan May, for his PhD work, brought ptarmigan down from the alpine and put them in heat chambers, and he warmed them, and he monitored their physiological and metabolic stresses and saw that they were essentially burning the brown fat equivalent that mammals do in birds once temperatures got above about 20, 21 degrees Celsius. That has huge implications because these birds are so well adapted to these cold environments. But in the summertime, when it gets hot, birds seek out these north-facing slopes or underneath boulders where temperatures can be 10, 20, 30 degrees cooler. And they also sneak out these permanent snow or ice fields to basically actively thermoregulate. What we've seen in the last 30 years is the number of days where temperatures exceed 21 degrees is drastically growing, and those ice fields have actually disappeared in the last 10 years in a lot of the alpine habitat. So with climate change, those fields are no longer permanent. Some of the thermal refugia that Janet referred to earlier with our ground squirrel examples are no longer available. How are these animals going to cope? How are they going to adapt if they can? So what we're seeing, next slide, is that, as I mentioned a bit earlier, we're seeing birds are trying to cope with this, but you've got a bird that goes through a nuptial molt in the spring, moving from its pure white plumage to try to match its environment. These advances are happening rather quickly. The molt is based on photoperiod in the days, and there may be mismatches where you have white birds living on brown landscapes, and they're severely more susceptible to predation events. We are seeing that those hatch dates are moving forward by as much as 2 to 4 days per decade, and we have 50 years of data, over 50 years of data looking at this, and birds are trying to match and meet that environment. But again, we don't know how plastic they are and how much they can evolve and continue into the future. And for ptarmigan, this is the classic example of the escalator to extinction, when if everything shifts upslope, there's only so much mountain left where they can persist.
Right, and sticking with the idea of color coat mismatch, there's actually a really great example and kind of a positive example for white-tailed jackrabbits across the western and central part of the US. And this was some work out of Scott Mills' lab at Montana. So white-tailed jackrabbits turn white in the winter, but not all of them. There are populations where individuals remain with a brown pelage throughout the winter. And this map shows the distribution of the species, and they used, I think, over 1,300 samples from museums to examine the coat color in the winter and then to map the probability of animals being brown versus white. And so that's the color ramp that you see across the distribution of the species. What's really interesting is that they were able to identify the fact that coat color change in this species is really only tied to 3 genes. And using a projecting the climate forward, they were able to document that there would be widespread mismatch with a reduction of snow cover going forward, kind of what you see in the bottom right there. And what's really sort of heartening about this example is they also conducted simulations to to look at the genetic rate of change for these populations that are mismatched. And those simulations suggest that populations should be able to evolve fairly rapidly to develop and retain a greater percentage of that brown color code during the winter when they have less snow on the ground. So it's a story about how species, if there is genetic variation in the species, in the population, they can adapt to these changes in ways that might allow them to persist even under sort of pretty remarkable changes in their environment.
Thanks, guys. So one of the other considerations for species response is that, species are not existing in isolation and that impacts to one species can have positive and negative impacts on other species in the community. And this is a paper that just came out this year that was investigating sort of the competitive dynamics between deer mice and Columbia Plateau pocket mice in the sagebrush. And they were interested in what happens when there's juniper encroachment and cheatgrass invasion in this system and how it impacts these competitive dynamics. And what they found is that at low levels of disturbance, deer mice really benefited and it expanded their home range, which caused competitive exclusion of these pocket mice. And in the discussion in the paper, they discussed how increasing deer mice populations can have negative impacts for brewer sparrow because they're a nest predator, but can have positive effects for other generalist species like common ravens. And so you can imagine as common ravens start to increase in abundance, then you can have even more interactions sort of cascading through the web there. So it's just one of many ways by which we can have these direct and indirect effects on species.
So we don't know a lot about the disease dynamics and the potential implications for climate change, but it's thought that within these increasing warming temperatures, there might be more risks associated with disease. As an example, back in the early 2000s, associated with sage grouse, we saw the sweep of West Nile virus moving from east to west across the conterminous US. And in 2003 and 4, where we tracked actually West Nile virus initially occurring in some of these westernmost states, we saw massive reductions of over 60% in survival of sage grouse populations as they were first exposed to West Nile virus. So this has huge implications. We've noted a bit more recently that West Nile isn't nearly as profound as it was when it was initially introduced to these populations, and there's maybe a small potential for a little bit of immunity. But as climates warm, we get the vector that transmits this disease, a mosquito primarily in the west called Culex tarsalis, can rapidly increase its breeding cycle. And as those populations increase, all they need is a hoof print of water to lay their eggs and reproduce. And with the sporadic nature of spring snowstorms or more moisture coming as precipitation as opposed to snow, it's warmer. There's the opportunity for things like West Nile virus to really ramp up and could pose more risks to wildlife populations. And you get what you see in the top right with just sage grouse that was dying of hemorrhagic fever from being exposed to West Nile virus.
All right, so there are a ton of changes, like you guys noted in that first poll, that we're already observing in the sagebrush system. And I want to switch now to talking about how we incorporate all of those observed changes and all the data we're gathering on species vulnerability into conservation and management plans. One of those ways is through climate change vulnerability assessments. And these are it's essentially a framework for integrating vulnerability information. So I'm just going to give a quick overview here of, what exactly do we mean by climate change vulnerability? You may be familiar with this Venn diagram here in the bottom right, which shows traditionally the three components of vulnerability, which are exposure, sensitivity, and adaptive capacity. And I just want to walk through sort of the mechanisms by which those relate and confer vulnerability. So exposure is the rate and magnitude of climate change that is experienced by the population or organism or the species as a whole. And exposure can be characterized as annual conditions, seasonal conditions, variability in those conditions. And we're really starting to shed light on the impacts of extreme events, as well as the velocity of change, and when and where we're exceeding sort of these thresholds of tolerance for species. And sensitivity, I like, there's some ambiguity between sensitivity and adaptive capacity. And I like to think of sensitivity sort of from a like pharmacology, toxicology perspective, and that it's the dose response or exposure response of the species. And that simply just means it's the degree of impact or susceptibility that the species may have to specific climate variables. So a species may be, in this example, more sensitive to extreme conditions or changes in extremes than they are to mean annual changes over X years. And so we may upweight the importance of those extreme conditions in our vulnerability assessment. And together, exposure and sensitivity combine to reflect the potential impact of climate change on the species. An adaptive capacity is this mediating factor. It is the species' ability to cope with or adjust to those changes. And when combined with that potential impact, that is the true or realized vulnerability of the species. And most of my work focuses on adaptive capacity. We've found that that's really that's really the one piece that managers can leverage for managing species when it comes to climate change vulnerability. And so we spent a lot of effort trying to operationalize that topic. And Eric Bieber and others came out with this great paper back in 2016 that really builds on our conceptual understanding of what is adaptive capacity. And what they showed is that species have this intrinsic ability to accommodate climate change. And that depends on their evolutionary potential and their plasticity, as well as some key life history traits and movement abilities. And they called this the fundamental adaptive capacity, relating it to that ecological niche concept. But then we have what we call realized adaptive capacity. And that's simply acknowledging that there are external factors, typically other anthropogenic stressors like barriers to movement, that can affect or constrain that species' fundamental adaptive capacity. So really, things can limit the full expression of species adaptive capacity. And so this is sort of our conceptual basis for what adaptive capacity is. And then a few years back, we decided to put together an assessment framework for evaluating species adaptive capacity. And we presented this framework through this lens of species' ability to either persist in place or shift in space. So species can either adapt in situ or acclimate where they currently exist, or if they have the capacity, they can move to track suitable climate through range shifting. And in some cases, unfortunately, we do see local extinction events. But we wanted to really dig into what kind of traits may confer greater adaptive capacity for a species. And there was some foundational work that's you can access through that link down there, where they discuss the kinds of traits that might confer greater adaptive capacity in species. And these are things like short generation time, so those short-lived species with high reproductive output, those ecological generalists that occur across broader spatial distributions, and that many of which have greater genetic diversity. And truly, what comes up a lot in my conversations with folks about these traits is that this sounds a lot like an invasive species. And in fact, many of these high AC characteristics are what we see in invasive species, or in generalist species like here, the raven. And so we synthesized from the literature all that we could about the kinds of traits that are used to measure adaptive capacity and sometimes vulnerability more broadly. And what we found was that there are 37 traits, or what we call attributes, that we grouped into 7 complexes. And we've actually added 2 new traits since the publishing of this paper back in 2020, which will be released soon. But so these traits are arrayed by those related to species distribution, those related to their movement, that's where all the migration attributes are, as well as their evolutionary potential, their ecological role, abiotic niche, life history, and demography. And we've spent also a considerable amount of effort focusing in on this evolutionary potential piece. And if you're interested, this paper discusses how you might measure evolutionary potential and what kind of proxies you can use to get at some of those key markers of evolutionary potential, or it's also referred to as evolutionary adaptive capacity. So I wanted to show you two examples of how we put this AC wheel into practice. And this is an example of an assessment that was completed for the greater sage grouse. And you can see here that same wheel of traits in the same order, but now they're color coded by that level of adaptive capacity, with the darker blue being higher AC and the deeper red being lower AC. And then the length of these spokes in the wheel represent the strength of evidence used in the strength of evidence for that assessment for that trait. So shorter spokes mean there's less certainty in that assessment. And then this inset figure here in the top right is just an arrayed version of the bigger wheel, ordered from high to low adaptive capacity and high to low strength of evidence. So sort of just a summary figure. And the sage grouse is a pretty well-studied species. So not a lot of information gaps there, but quite a bit of low adaptive capacity attributes spread throughout this wheel. And we've also done a similar assessment for the pygmy rabbit. So same setup here. And just one of the really interesting things that I take home from this assessment is that this is more of what I would consider a data deficient species, and that there are some really obvious information gaps that could be filled to support our understanding of the species adaptive capacity. And that is any and everything related to their abiotic niche. So their behavioral flexibility, their physiological tolerances, their climatic niche, and phenology, excuse me, as well as their demographic traits. So quite a bit to learn from this species to support our understanding of its adaptive capacity. And if you're interested, we do have a quick reference guide for doing these adaptive capacity assessments. The species that you work on may have already been done. We've done quite a bit of Western species already. We have 70 assessments that are more or less complete, and we've got 500 more planned by 2025 as part of State Wildlife Action Plan revisions. So in this little tiny URL here, we do have a spreadsheet that lists out all the species that are completed and ongoing and planned, if you want to use that as a reference. We also have a lot of guidance out there, not only about adaptive capacity, but climate change vulnerability assessments in general. And if you're interested in those, I did help work on this voluntary guidance for states, for state wildlife action plans. We're in the process of updating the National Fish, Wildlife, and Plants Climate Adaptation Strategy. And then there's specific guidance that we developed for linking adaptive capacity assessments to species status assessments, which many of you with Fish and Wildlife Service might be interested in. And then we're also updating NatureServe's Climate Change Vulnerability Index, bringing that online and into a web-hosted GIS-based platform that will now include all of these AC traits. So there's lots to come, and we'll cover much more of this in the in-person workshop for those that are attending. And I'll turn it over to Cam now for a great example of AC.
Thanks. So I'm going to walk you through a real-world example of adaptive capacity, leaning again on our white-tailed ptarmigan examples. So this is using a genetic basis for white-tailed ptarmigan. We've assessed the subspecies designations, which you see the 5 located here in the different colors that are represented. And we are really interested in assessing how genetic variation might be adapted to local environmental conditions, if there was any evidence of that adaptive divergence. So there's multiple environmental gradients, which I'm showing on the right here, that many link to climate, but it changes in rainfall, snowfall, temperature, obviously elevation, and then some habitats conditions. So there are multiple environmental gradients across the range as well in the southern subspecies, and we use SNP, single nucleotide polymorphism data, that we could use for birds across the entire range. we developed a partial redundancy analysis, but basically it's an ordination approach that we can assign relationships to the genetic information and the environmental conditions. And we're trying to characterize if those environmental gradients can actually identify potential loci that are under selection. And we've identified that link those to the nearest gene that's assumed to be linked with the candidate adaptive SNPs. Next slide. So what we see is that we found there is indeed adaptive divergence patterns associated with combinations of local vegetation characteristics, phenological characteristics, precipitation, elevation, latitude, etc. And there's a significant association with genes in a functional group called the EGF domain, which is listed there. It suggests that there's different adaptations for local diet, obviously because morphology changes for this species across elevation and latitudinal gradients. And oxygen levels, when you go higher elevation, there are much lower, so birds are adapted to those altitudes. And then obviously the phenological aspects, we talked about the timing of molt for these birds. So it's extremely important, given we already know that climate change has affected breeding phenology for white-tailed ptarmigan. And if it pushes these things further, do birds have the genetic basis to be able to adapt to those changing conditions? So if we take a zoomed in look on the next slide a little bit further down to the southern Adaptation subspecies, it occurs in Colorado and into New Mexico. We found precipitation and temperature gradients, even just within that smaller subset of range for the subspecies, was associated with that adaptive divergence. As climate continues to warm and alpine habitat is expected to be warmer and wetter being more rain as opposed to snowier, there's some concern over how birds are going to be able to adapt. So understanding where there's genetic variation associated with these local environmental conditions and considering what environment that they're in may help us predict parts of the range that might be more likely to persist into the future given continued climate change, or areas where the distribution is likely to shift. And then also help us with thinking about, do we need to consider assisted migrations? Are there areas in these populations that do not have the genetic basis to adapt or the subspecies to cope with a warming climate? And do we need to assist those by moving birds in to get those genes into those populations?
Right, thank you. So, yeah, I loved that example. I think it was a really great example of using genetic information to support our understanding of species adaptive capacity. And I want to take a step back now to think about the broader picture of vulnerability assessments and how we incorporate all these different threads of information. And there's really 3 different approaches to doing vulnerability assessment. Of course, there's variations or combinations of these, but typically vulnerability assessments are done using either a correlative, a mechanistic, or a trait-based approach. And correlative approaches are what we see with a lot of range shift modeling and species distribution modeling. And these are, essentially correlative relationships between species occurrences and underlying, environmental and climatic conditions. And this is a cool example here of sagebrush ecosystems in the Four Corners and Upper Rio Grande of the Southern Rockies. And they essentially mapped out vulnerability of sagebrush in this system. And you can kind of see those hot spots of vulnerability in the darker red. Mechanistic approaches focus more so on like the underlying processes through which climate change can impact species or even habitats. And oftentimes these incorporate other stressors into these models. But they're really helpful with revealing important insights about underlying ecoevolutionary processes, for example, although they can be difficult to parameterize and are certainly more computationally intensive particularly over larger extents than these other 2 approaches. And this is an example here of, modeling demographic change in, I forget which species this was, but they were essentially looking at recovery after disturbance and found that essentially this population didn't show great rebound after disturbance, and so they were sort of linking, you know, those environmental parameters to this rate of recovery. And this can be incorporated into vulnerability assessments. The other option, which I'm more familiar with, is this trait-based approach. And this is just an example here of one of the AC wheels. You know, NatureServe's Climate Change Vulnerability Index is a good example of a trait-based approach. And these are often sort of an integration of mechanistic information and correlative information, and usually are done through expert elicitation processes. And so, you know, one of the questions we can ask is like, why do a vulnerability assessment? How does this help us in practice? And I thought I'd use this climate adaptation cycle to illustrate the many ways in which vulnerability assessments can be super useful for conservation and management, planning and decision-making and implementation. And in this climate adaptation cycle, step one is usually to define your planning purpose and scope. And this is the same for any management or adaptive management process. And vulnerability assessments are one way to really hone in and address very location or context-specific management concerns. And when doing the vulnerability assessment, you can ask questions related to what's at risk, which species are at risk, why are they at risk, and to what are they most at risk of, or what climate variable threatens them the most. Where are they most at risk? You know, that is a common question we ask when doing those correlative species distribution model type vulnerability assessments. And in some instances, we can often get at the when question. So when should we be most concerned about these impacts manifesting? And that's where those mechanistic assessments often come into play. And through this process, we use that information to review and revise our goals and objectives and identify adaptation or management actions. And these vulnerability assessments are great risk assessment frameworks for then helping us to inform how we can best triage those priorities that we have, those specific goals and objectives that we've identified. And, you know, through this process, selecting the actions that we wish to implement. And then lastly, that vulnerability information can also help us more effectively and efficiently allocate limited resources. So it hones in on the what, why, where, and when, and helps us to, you know, select and implement the most effective actions. And of course, through a learning and adaptive management cycle, we iterate this process and learn from these adaptation experiments. But there's just many ways. Essentially what I'm trying to show here is that there's many ways that vulnerability information can be leveraged in decision-making. And for the last bit of our presentation here, we've got two case studies that Cam and Janet are going to share with us. And I will stop sharing my screen because they're going to take over with their slides.
Great. Cam, did we decide I'm going first?
I think so.
I think so.
Okay. Can you guys see that?
Yep.
Okay, great. So I'm going to give a little bit of an example with pygmy rabbits. And I think Lindsay really introduced the idea that we have a lot of gaps in understanding about their ecology and how they might respond to climate change. So I'm going to just give a little bit of an overview of some work that we've done, some questions that we have, and work that still needs to be done to address some of these major components of adaptive capacity. So I'll talk a little bit about distribution and potential shifts in distribution under climate change. I will talk a little bit about that behavioral plasticity, and then I'll end with some work that deals with adaptation. So what you need to know about pygmy rabbits are really two things. They're sagebrush obligates, but sagebrush is really important for a variety of functions. So in the winter, something like 98% of their diet is sagebrush. In the summer, it's about 50%. But sagebrush is really key. They only occur in sagebrush environments. The other thing that's a little bit unique about them is that they're obligate burrowers. So they create and use residential burrow systems year-round. They are always associated with those burrow systems. And yet it's interesting that there are other lagomorphs that share the same environment, cottontails, jackrabbits, that are not obligate burrowers. So there have been a number of ideas about why pygmy rabbits have this obligate burrowing behavior. To advance this, there we go. Okay, so a little bit about distribution. So this is a figure actually from the Federal Register. It was after one of the first rounds of petitioning the species for endangered or threatened status throughout their range. And this was this was presented as the historical and the current range of the species. And one thing I think it's really important to distinguish is a range versus a distribution. So this is an outline of the extent of the area occupied, but it doesn't really tell us anything about the distribution within that outline. And I should also just preface that this also doesn't include the Washington distinct population that's in the Columbia Basin. So one of the first things we wanted to do was to try and understand something about the distribution so we would have a baseline against which we could measure changes in distribution over time. And so we gathered known occurrences. There were about, I think, 2,000 occurrences that we could confirm from throughout the species range. And those are shown here on the right. And we used that information to create a species distribution model. And that work was some work that was done by one of my graduate students, Ian Smith, and also collaborators, Leona Swansara and Sonia Knedder. At that time, both of them were with Idaho Department of Fish and Game. And so this map shows the outcome of that, that modeling effort. So we identified over 14 million hectares of what we called primary habitat. And those are habitats that are most similar to occupied areas. And I should say that we used habitat variables included vegetation, topography, and also climate variables. One of the things that I found really surprising in looking at this map once we finished it was the fact that there are these sort of 4 core areas that have sort of areas, relatively large areas of contiguous habitat, but they're separated here by areas of where the species doesn't occur and where their habitat is not predicted to occur. Not dissimilar from sage-grouse and probably not surprising. Now, developing this type of a model, it's useful at the range extent, but we know that these types of models tend to overpredict occurrence. And within the state of Idaho, there was an interest in using these models to try and cite energy infrastructure, roads, fire breaks. And it felt really bad about saying, yeah, we did all this modeling work, but it's really not very useful for what you want. And to address that, had another graduate student, Lindsay Rush, and she developed models, similar approach to developing species distribution models, but this time within 5 different regions within Idaho. So just to orient you, this is where we are. And these are pretty different, these areas have pretty different climates, from down in the Owahis where it's really dry and hot, over here in the Bear Lake Plateau, fairly cool, higher elevation up here in these mountain valleys. So this map shows the primary habitat, this time in red, and then secondary or suitable habitat in pink. And as we expected, it predicted quite a bit less habitat. So there was about 80% less primary habitat than what we would predict over the same region for the range-wide model. One of the things that came out, a couple of things that came out of this that were really interesting, is that the habitat associations really differed across all of these regions, these 5 regions, and that the bioclimatic variables were really strong predictors. In fact, they were the strongest variables in the models. And so this got us thinking, like, could we just use these models to project forward and ask what, under different forecasted climate scenarios, where do we expect the environment, the climatic envelopes to be in the future? And we did that using a couple of different climate projections. And this, I'm going to show you two different maps. This first one here is a sort of more moderate climate projection. This time the red refers to areas that of contraction, so areas that were suitable habitat that are predicted to no longer be suitable habitat in the future. The blue are colors that were suitable habitat that are predicted to persist as suitable habitat, and then the green represents areas of expansion that may become suitable in the future. And we did that under a more moderate climate forecast and also under a more larger response. And the patterns are pretty much the same, except down here at Bear Lake, I don't know, they're going to be knee deep in rabbits, apparently. Now, there's lots of reasons why these models are really just a first cut. They don't really represent reality. They assume that there's sort of a stationarity in the relationship between the animals and the environment. They also don't incorporate any of the information about biological responses, changes in their physiology, their behavior, or the ability to adapt. And so right now we are working on refining some of this using an occupancy modeling approach that will allow us to get at a dynamic changes in the occupancy over time, not just the distribution of potential habitat. And this is a really large collaborative effort with Idaho Department of Fish and Game, Montana Fish, Wildlife, and Parks, and it's being led by a graduate student here, Fiona McGibbon. And we are conducting winter surveys. We're collecting fecal pellets to identify species, to confirm species and separate pygmy rabbits from prints that might be made by other lagomorphs. And just to show you where we are, these are the results as of the end of February this year. So the red dots are sites where we believe we detected pygmy rabbits based on the pellets not yet confirmed with the genetic analyses, places where there was no detection, and places that are not surveyed. So like I said, this is a really large collaborative effort. We had over 80 people out helping with these surveys. And so here's a shameless plug. If anybody would like to come out next winter and help us with those surveys, we're hoping to get up to something in the number of 800. So yeah, if you're looking for an excuse to get out of the office in the winter, we would love the help. But we'll be using the results of this to get a better understanding of changes in distribution and the environmental factors that are associated with occupancy. Okay, I'm going to switch gears and talk a little bit about behavioral plasticity. And I talked earlier about the Arctic ground squirrels. Pygmy rabbits are obligate burrowers. They use burrows. There's different ideas about why they use burrows. One of the ideas is that those burrows serve as thermal refuges. And so several years ago, one of my PhD students, Charlotte Milling, tried to address this question and try to understand the degree to which burrows actually function as thermal refuges, both during the summer and the winter. And to do this, we measured the temperature in the burrows, and we also measured the environmental temperature at a lot of microsites above ground where animals could rest. But what's important is that this environmental temperature is not ambient temperature, it's not air temperature, it's actually measured using models that mimic the way that animals exchange heat. And notably, these are models that warm up in the sun, so they incorporate that radiant heat. And they're actually very fancy copper toilet floats is what they really are, that had a thermocouple inside. And we compared these temperatures. We use this to map throughout the summer and the winter when those temperatures were within the thermal neutral zone of the animals. To evaluate energy expenditure and thermal neutral zone, we also did work with captive animals and respirometry. And this is a way to measure oxygen consumption, so we can actually measure the energy that is being used or model the energy that's being used at different temperatures. And the outcome, when you ask, was the burrow actually a thermal refuge? In the winter, it was. Usually on average about half the day, about 13 hours of most days, about 56% of the time, being in the burrow was more energetically beneficial than any place on the surface during the winter. During the summer, however, the story was a little bit different. Only for 2.5 to 3 hours a day, about 10% of the time, was the environment, the temperature within the burrow system actually a refuge for them. And that sounds really counterintuitive, and it's not what we expected. The reason for that is that the temperature in the burrow is cold, and it's below their thermal neutral zone, which means when they go in the burrow system, they have to crank up their metabolism in order to stay warm. And the most thermally beneficial or neutral places for them turned out to be on the surface, out of the sun, under the shade of sagebrush. And so the question is, given this, I mean, maybe burrows still are refuges in the summer and could serve as a thermal refuge if rabbits can accomplish this sort of shuttling thermal regulation that I described earlier for the Arctic ground squirrels. We're not sure they really can, and a couple of reasons. One, we haven't documented it, although we're going to be looking for it. But another is that loporids, rabbits and hares, lack some of the brain structure, the structure of the blood vessels around the brain that facilitate brain cooling. So in order to be able to take advantage of shuttling thermal regulation, animals need to be able to build up a body heat to maintain that body heat and then go underground or to another refuge and shed that. And we're not sure that rabbits can, but so we don't know. Rabbits in general tend to be pretty vulnerable to warm temperatures, surprisingly more so than cold temperatures. Okay, I am just going to end with saying that I wish I had all the data that Cam has for his ptarmigan and the data they have for grouse. We are just beginning a project this fall to examine adaptive genomic variation to try and understand whether or not there are traits associated with energetics, with thermal tolerance, and whether or not we can detect those differences throughout the range of the species. And even though, I mean, to a lot of us, sagebrush looks like sagebrush, across this broad range of the distribution of pygmy rabbits, there's actually a broad range of environments. So this is something that we are just starting this fall, that's in collaboration with Lisette Waits and Paul Hohenlohe, and also some folks in Wyoming and Nevada. So we're excited to be able to have the type of data that you guys have for grouse and for ptarmigan. So that's what we know for pygmy rabbits and adaptive capacity. And I think there is a lot of information yet to be learned about this species. And this is our framework for starting to address those questions. So I'm going to go ahead and stop sharing there and hand it off to you, Cam.
Thanks. Okay, are you guys able to see my first slide?
Yeah.
Great. So I'm going to shift focus, but stick within the sagebrush obligates. We're going to go from a mammal to a bird. I'm not going to give much background on sage grouse because I think everybody's fairly familiar with them, but there's two species of sage grouse. I'll talk a bit about greater sage grouse today. I'm going to talk more generally about them, about possible effects to sage grouse, generally both species, and then a bit on some greater sage grouse trends and how we might think about climate links. There's obviously lots of studies out there. Then I'll shift focus and talk a little bit about Gunnison sage grouse, and specifically a climate vulnerability assessment that we just published a month ago for Gunnison sage grouse. And then I'm going to come back to, much like the ptarmigan work, and lean on some of our work with Sarah McCants and Shauna Zimmerman, looking at genetic adaptive capacity for sage grouse as a whole. So we've talked about a lot about direct and indirect effects, and this is a nice graphic that Carl Lundblad's prepared for a wildlife monograph manuscript, synthesizing some of the effects of climate on sage grouse in general. Direct effects are hard to track, though we do see in a lot of different cases there can be direct consequences for sage grouse: exposure, nest failure due to freezing, wet changes in climate with extreme spring temperatures causing snowstorm events and how it can have implications for reproduction. I'm going to focus a little bit more on the shift all the way over to the right side where we're talking about some of these indirect effects. First, I'll start with the second figure that Carl's produced for that same manuscript that is sort of characterize some of these indirect effects on sage grouse. We know precipitation is going to change. We're losing some snow, which is extremely important in a lot of these systems with winter moisture coming that percolates into the soils. As that changes to rain tends to run off. We don't get it percolating into the soils. We have lower soil moisture. Temperatures warm those up. We have more evapotranspiration that results in less moisture availability. If you're looking over here towards the herbaceous production, which is key, I mentioned this for ptarmigan, for food resources for sage grouse chicks, as well as the insects that are provided by those. That can also result in reductions in concealment cover that allows birds to hide, escape predators, and protect nests, and probably also some thermal issues with nest sites. Combined, we can see that the lack of soil moisture, increasing temperatures. We've all heard about the exotic annual grass cycles and cheatgrass moving in, less moisture, fires burn hotter, cheatgrass, the annual grasses take over, provide more fuels, and we have direct losses of habitat, which pose huge consequences and one of the biggest threats to sage grouse and their ecosystem. We know that sage grouse populations cycle over time, and this is just a graph going back from the 60s today, and we tend to see these oscillations that occur and continue to occur over time. What drives those is we've shown in various different places across the range that it is sort of tied to climatic patterns. And as those shift, there could be consequences for how populations may also shift or change, or abundances may track. This is data from let counts compiled from across the entire range, a project that Pete Coates and I lead. Just draw your attention to an article that Pete and his shop published a few years ago in 2018, showing how there's in the bi-state population of sage grouse, there is some direct links with precipitation and how we see the slight lag effects possibly with regional population trends associated with sage grouse populations. So we know climate can have impacts in these populations, mainly through habitat mediated consequences and resources that are available. So we've developed this larger population framework, and this is just to show an example of how we tend to see these populations and natural trends. If a population was stable over time, we'd see these stochastic factors such as climate that may drive these cycles. And in this case, we might have a constant carrying capacity where populations just bounce up and down around this carrying capacity. But that's not the case with sage grouse. We tend to see these declines that are happening. So not only are these oscillations driven by stochastic climatic factors, but there's other deterministic factors or stressors, fires, loss of habitat. Those can be also induced by climate-induced changes, increasing the fire frequencies. We're losing habitat. We're seeing population numbers decline, and climate can have direct effects on that. What we're seeing is carrying capacity has dropped. We've got smaller amounts of habitat, lower quality, and less birds are able to be supported by those habitats. We've characterized all of this in a large monitoring framework. And I won't go into details here, but just to show that we can model trends and patterns in those trends across the entire range, across distinct population units. And what we're doing right now is assessing a variety of different factors that may be related to those population trends, including climatic conditions. So I'm going to switch right now and talk a little bit about Gunnison sage grouse and talk about this vulnerability assessment and look at that for sage grouse that we just recently published. Gunnison sage grouse occur in southwest Colorado, as you see in this map, largest population in the Gunnison Basin, with distinct small satellite populations further to the west and into Utah. There's maybe 3,000 to 5,000 birds remaining for this species. They are a threatened species. And the majority of those birds, 85%, occur within the Gunnison Basin population. So following in line with the species status assessment that was recently completed, we set out to develop 3 different scenarios following within the species status assessment, looking at optimistic continuation and pessimistic's cutoff that under RPC 4.5 and 8.5 scenarios. So these generally relate to warmer, wetter, warmer, drier sort of scenarios. We created these projections for each population based on 10 data sets of current and projected to 2070 threats to Gunnison sage grouse habitat. And these focus on potential loss of sagebrush range, where that has occurred and we'll lose sagebrush cover, drying of mesic habitats across the populations, expansion of pinyon juniper encroachment, particularly with climate change, and also invasive annual grasses. We also assess development, future development risk, as well as wildfire risk, but I won't go into those today since we're focusing primarily on climate change. So what we found is we can characterize these different patterns for each individual population, which is shown here. The Gunnison population is shown right here, and the other 6 satellite populations are shown listed here. So red here shows a fully degraded non-habitat current today, what's available within these populations, and this is focused just on sagebrush. And then the tan colors represent that they're sometimes at risk into the future or always at risk into the future. And then the blue color represents that it's stable habitat going into the future. So from a climate perspective, we're seeing really today, some of these populations have really degraded sagebrush habitat already. The Gunnison population, which is the stronghold, is probably at its best right now. And the threats to losses of sagebrush aren't huge, with the exception of maybe in the San Miguel Basin population looking forward. However, we can look at through the other conditions, the mesic resources, which I show up here on the right. You can see the majority of these populations are dry, they've lost mesic resources, and they're at least sometimes, if not always, at risk for losing those into the future in 2070. The same goes for the invasive plants, with pinyon juniper being, native creeping down, exotic invasive annual grasses. Most of these populations are at high risk of some of these exotic invasive plants. And that also is going to bring higher risk of fire potential, which I'm not showing here, but we did characterize. So it doesn't bode terribly well for these populations, but we can map these out with these risks. And what I'm showing here is the mean overall vulnerability score combined across the 6 different factors. I'm showing you the optimistic, so we're reducing actually our warming, our continuation as is, and the pessimistic with the 8.5 scenario. Draw your attention to the Cerro Sims Summit population right here. You can see in all 3 scenarios, this has the highest overall vulnerability with a lot of risk factors that are happening here. We also see here in this Dove Creek population Dry Creek of the San Miguel Basin population is heavily at risk as well. And this is owing to losses of sagebrush and high fire risk associated with this. What also draws that attention to is that overall in the optimistic and continuation scenarios, there's pretty low risk to the Gunnison Basin population, which is pretty positive and optimistic. If you look at the pessimistic scenarios there, that's where we start to see some things creeping in here. And this is the hot, dry future climate scenario, which results in a much greater exposure and a lot of loss of habitat, primarily due to pinyon juniper in these hot, dry scenarios encroaching, as well as some of the invasive annual grasses taking a better stronghold. So I'm going to switch my last little piece here to talk about genome-wide divergence for greater sagebrush. And as I mentioned, this is work that Shauna Zimmerman is now leading with Sarah Oler-Marcantz, who's a great molecular ecologist here at Fort Collins Science Center. So we know that sagebrush community is variable across the range. Janet alluded to that in some of her last slides. There's distinct compositions of sagebrush and shrub communities and subspecies, and they vary in their compositions of concentrations of different chemicals and those toxins that are used to deter or defend off herbivores. This really raises the question about local adaptation for sage grouse, particularly for things like winter diet and the relative ability of these different populations to deal with those different toxins. We also know that temperature and precipitation gradients exist across the sagebrush biome. That's kind of what drives why those differences in species and subspecies of sagebrush exist. But we can explore the potential for local adaptation to diet and to environmental gradients. Kevin O was the first to develop to lead work specifically looking at the questions of divergence among toxins and tolerance genes across the sagebrush distribution. He developed the first sage grouse reference genome, and then for greater sage grouse and Gunnison sage grouse. And Shauna has since expanded on this work, but originally we found there's strong divergence associated with the cytochrome P450 genes. And these and other genes are known to be significant detoxification in the detoxification of plant secondary metabolites, or PSMs, and those toxins that sagebrush plants produce. But each species and subspecies of sagebrush produces a unique set of different toxins. So Shauna also found some similar relationships for Gunnison sage-grouse across environmental conditions, and they're variable with the sagebrush habitat compositions. So we've since developed a complete annotated genome for sage-grouse. We expanded that genomic data set with these samples from these additional sites. And this is allowing us to better characterize the relationship between adaptation in the sagebrush community and the local climate. So we might provide some insight into the common management interventions, such as restoration of habitats. If we're going to restore habitats, we better have the right sagebrush that sage-grouse are adapted to deal with those toxins. If not, sage-grouse better have the adaptive capacity to express different genes to deal with those different toxins that are associated with those specific habitat types. That has huge implications for translocation of sage-grouse, as well as for climate change. As climate tends to shift, we're going to see transitions. We need to make sure that sage-grouse are occurring in the right places that they're adapted to, or ensure that they have the right genes to be able to deal in those particular populations. So range-wide, we see that there is population structure for sage-grouse, and it's different across these areas. And what we can do is we can take the adaptation that sage-grouse have, and we can make climate models to forecast how these genes, the climate envelope conditions of where these genes are adapted to cope with. And we can see that with these gradient across the range from high seasonal temperatures in the north to low seasonal temperatures in the southwest. And we can see this adaptive index, and we can see that's really sage-grouse in the north and the east are really genetically adapted to deal with that environmental gradient of those different types of climatic conditions. And what we can do with this is we can actually forecast this to the future based on what climate conditions are look like. And we're going to look at the separation, or how far away the genetic adaptation locally is to the climate that's going to be in the future. And this is this genomic offset using another RDA type of analysis, this ordination approaches, and we can see how far we are going to be off given future climatic conditions and what they're predicted to look like. So these areas in brown are going to have much higher differences from the genetic basis of what birds are able to deal with the current climatic conditions. So this is essentially a genetic offset map where you can assess these divergences. It means birds will need to be genetically very well adapted to the future climate conditions, so they're going to be further off. So this could have real insights into how we might manage the landscapes, considering sagebrush and considering the birds. So I'm going to wrap and say, just come back to the fact that climate change is happening. We're seeing shifts in the ecosystems, the sagebrush, the habitat, the characteristics. We're seeing changes with exotic invasives, fire regimes, the temperature, precipitation, the soil moistures. We're going to see changes to the resource and the conditions in the sagebrush ecosystems that out there. The species that rely on these are either going to have to adapt, like we've talked about with the genetic capacity, if they can or not, but the conditions are going to shift. We're going to go from a larger intact sagebrush with decent herbaceous and perennial understory components to maybe looking a little bit depauperate. What we want to do is resist that change that's going to transition to a sea of cheatgrass where many species cannot make a living. But we might also have to accept that the sagebrush of tomorrow is going to look a little bit different, and we have to try to understand how species like sage-grouse and pygmy rabbits are going to be able to adapt and persist in those landscapes into the future. So I'll stop.
And I want to say thank you again to all of our incredible presenters. It was really great to learn from you today.